I'm going to talk about preterm birth, and I'm going to talk about male-female ratios as it relates to preterm birth. I'm going to present some preliminary studies and results. These are all unpublished at this point. So I'm a perinatal epidemiologist. The best I'm going to do is some musings on mechanisms. I'm not going to make strong inferences. I'll let my colleague Brice come up and make some strong causal things if he wants to. You know, biologists are more apt to make mechanistic theories come true. So preterm birth looks a little like this. So why preterm birth? So babies born even a few weeks too soon can face a variety of morbidities and mortality. The morbidities focusing around respiratory problems, underdeveloped organs, cerebral palsy, certainly potentially life-threatening infections, and lifelong disabilities. So preterm birth is important. In this country, preterm birth affects about 1 in 8 babies born. It's the leading cause of newborn death in the US. The US has one of the higher preterm birth rates in the world. So keep that in mind if you're a human biology major. This is a very important problem. Think about it, 1 in 8 babies born. The March of Dimes has told us that the costs associated with preterm birth exceed $26 billion on an annual basis. And one of the conundrums in all of this is that preterm birth has been going up since the 1980s. It took a short time where it flattened out about 2 years ago, 3 years ago, and looked like it was going to go down. It's not doing that anymore. It continues to rise. So the March of Dimes, in their wisdom about 8 years ago, decided to make a 10-year commitment to David Stevenson and a few of his colleagues, including me, to establish what is referred to as a transdisciplinary center here at Stanford. We created that. We were much younger then, or it's made us look much older. I'm not sure. This transdisciplinary center is doing incredibly well. It has more than 100 investigators involved in doing a variety of things. The way I describe sort of my work in the transdisciplinary center, on one moment we may be talking about what Brice will show you, and that is sort of a single cell signaling on the surface of a cell, all the way to counting the number of liquor stores on a particular corner in a particular location as it relates to the risk of preterm birth. So if you can imagine that range of sort of thinking, that's what the Transdisciplinary Center does. It's very heavy on omics. We have made great strides here on this campus with respect to understanding the transcriptome, the genome, the microbiome, the proteome, and the immunome as it relates to preterm birth. I'm not talking about any of that. Brice will pick up a little bit of the immunome as it relates to CyTOF. I'm going to give you a straightforward epidemiologic kind of presentation with data. So you already heard that preterm birth is, let me set some ground rules. Preterm birth is defined as less than 37 weeks. Time is not akin to a definition of disease. So when we think about preterm birth, we have to think about much finer definitions within the rubric of this less than 37 weeks. So that's just something to keep in mind. It'd be akin to thinking about studying time to death in adulthood. So, but think about it in the inverse. It's time to birth from conception. So that's the way to think about it. It's not going to be one disease. There are multiple types of preterm birth. What I'm going to be talking about is spontaneous preterm birth, which represents somewhere around 50% of all preterm births, perhaps more. I'm not going to be talking about medically indicated preterm birth, which is generally comprised of preeclampsia and some other conditions. So, what's the epidemiology of preterm birth in one slide? Obesity, and I could talk about anything, any one of these is an entire talk for an hour, and I'm not going to do that obviously, but obesity is associated with preterm birth. So is low weight. So if you think about a shape of a curve in terms of risk, it's a J shape. The lower weight, normal weight, risk goes up with obesity. Independent of obesity, we also have demonstrated that short height is associated with preterm birth. Several of us have been working in the Central Valley of California for many years and have demonstrated a variety of air pollutants, mostly related to traffic emissions, are associated with preterm birth. We think this is mediated through an inflammatory response. I'd like to be able to stand up here and tell you that I've identified a variety of pesticides that are associated with preterm birth and other outcomes of pregnancy, and I can tell you that it goes the other way. They're protective. And I know I'm being filmed. I do not work for Monsanto or for any of the big chemical conglomerates, but I have a completely different take on that, and that is it's probably culling pregnancies that are at risk. Genetics. So you've heard folks already talk a little bit about schizophrenia and other adulthood and early childhood genetic phenomena being associated with conception and gestation. Preterm birth, so far people have looked at various GWAS studies and have identified small risks associated with certain genetic loci. Small risks like 1.2, 1.1, yes, they're statistically significant because they're based on such large numbers. The running theory is that preterm birth is maternally derived, and that's based on Scandinavian studies where it seems to be the frequency of preterm birth seems to be running through the maternal side of families. However, I think the jury's still out on the genetics. We published a little over a year ago, and this will be pertinent to our keynote speaker's talk and to what Amy presented, and that is that we looked at 800 trios, whole genome sequence data with respect to preterm birth, and identified de novo variants were associated with early brain programming. And they were literally abnormalities. That's what we annotated them to. So in other words, are we observing sort of a late miscarriage, if you will? That perhaps these babies should have been miscarried sooner, and they tended to be more in males. So that's just something to put out there. We've also observed that really young moms, let's say 13 to 16, are at greater risk for preterm birth. We've also observed that interpregnancy intervals, that is the time from the delivery of one pregnancy to the conception of the next, being less than 6 months, has been associated with an increased risk of preterm birth. All of these things explain a relatively small proportion of preterm birth. It's a complex disease. The most notable risk factor for preterm birth is black race in the US. So this is a slide graphic that was appeared in a New England Journal of Medicine. You can see here's the climbing trend. Here's births to all births, the climbing trend of preterm birth across time. This only goes up to 2006. It kind of continues to go like this, dropped a little bit and continues to rise. But the big separation is really between whites and blacks. It's about double for preterm birth overall, and it's at least triple for early preterm births. So that's a conundrum. And I can tell you that people, and we have too, have been trying to get underneath what that means, whether it's social disparity in terms of education, location, racism, on and on and on. We cannot, it's just impenetrable to understand what that is. So we've taken a few different approaches. It's not that we've given up on that. we've also looked at mixed race, and I'll come back to that in a few slides from now to show you sort of what we've found with that in terms of preterm birth. But where I really want to go is I want to show you male-female ratios. It's the centerpiece of what I want to talk about. So in California, for those who aren't familiar with what we have available to us here, it's really our community laboratory, if you will. There are 500,000 births a year. It's 1 in 8 of all the U.S. births right here under our study. 50,000 preterm births in this state alone every year. It's a diverse population, as most of you know, and we have many, many data resources, none of which I'm going to talk about, but all of which we use. So one of the primary uses of this laboratory are the administrative data that we have access to. So what I'm going to show you is the hospital discharge data. So this is all inpatient hospitalizations from 1990 to current. It involves 10 million births, so that's the analytic substrate of the results that I'm going to show you. Let me orient you to this slide. So I just want you to look at these two columns for now. So the sex ratio in term births is about 1.04. To tell you the truth, that's what everybody sees, no matter where you look. You look all around the US, that's what people see when it's all combined. In preterm birth defined overall as less than 37 weeks, we see it's right around 1.18. Male, an excess of males. Now look at when we define gestational age groupings a little bit finer. You can see this nice gradation. So the earlier the preterm birth, the more males. That's also seen, been observed, been published. So we said, okay, well, we looked at it again. What we wanted to know are there actually any patterns or elements of that sex ratio that might give us clues to preterm birth in general, and perhaps to the black-white disparity in particular. One of the things we did a couple of years ago was to see, did our air pollution results show different sort of effects on early preterm males versus early preterm females? The answer to that was no, but not surprisingly, because we had relatively small numbers. At any rate, we asked the question. I'm going to show you a series of slides like this, so let me orient you first so that it doesn't just look like a bunch of data in front of you. So here are the term births. Here's that ratio that I was talking about. So 1.04 in white non-Hispanics, 1.03 in Hispanics, 1.06 in Asians. So it's hovering in the 1.03 to 1.06. That's sort of what we expect. And here's that gradation that you saw. It bounces around, but these numbers are sizable, as you can see. I mean, we're talking very large numbers here. So it's higher in early gestations. Here are the gestations, and that's true for these 3 race ethnic groups. Everybody see that? Okay, I'm going to put blacks up here. It's going to change. So now focus on blacks. We have no idea why. I'm going to stop. I can stop right there. I don't have an answer. I really don't. But we're after this because we think this is, I mean, I can hypothesize a ton, but this is a robust finding, and I'm about to show you a series of slides where it doesn't change based on various strata. So let's take a look at maternal education. Is this a socioeconomic phenomenon based on education? The answer is no. It bounces around a little bit, but it's probably due to relatively small numbers in some of these areas. But these differences are still going to be statistically significant. It bounces just a little bit. I told you about, we had looked at obesity. There's a theory out there that obesity might contribute to the male-female ratio as well. A very elegant theory, actually. And I can get into that in more detail if you want. But we looked at that. Obese moms, is it different for obese moms? Nope. How about normal BMI moms? Nope. that ratio for blacks does not change across gestational age, while it stays the same for the others. What if we add, so I was showing you just live births. What if we add stillbirths into this mix? So stillbirths are defined as those that survived to 20 weeks but died after that. Could have been at any gestational age. So they got added in. And this is what surprised me, because we've actually published some material on stillbirths being a competing risk for, and there is a male-female ratio differential for stillbirths. I actually thought once we did this analysis, it would explain it, and it does not. So like I said, I don't have an answer. I really don't. So I mentioned earlier that we looked at the pairings of races, so white mother, white father, black mother, black father. And when we did that in the original analysis, what we saw was the preterm birth rate so all preterm birth, so if you had a black mother, black father versus this pairing, the odds ratio was 1.7, and you can see it was substantially higher in the earlier gestational weeks. So we theorized before doing the analysis that maybe what we could see was that it could be a specific pairing of the mother or father that was contributing to the male-female ratio. So let me go slow with this because it took me a while to grasp this. So here's the white-white pairing. There's this ratio once again. That's the ratio that we've been looking at. The same male-female ratio. It's 1.05 or so in term, and it's higher in the early gestational ages. Now let's add the black mother-father, and there's the ratio that we've been seeing. It doesn't change that much as you go across. Anybody want to take a guess what might happen here? What? Who said what? What did you say? So it looks like, you might walk away from this and say, maybe the mother is contributing a little bit more than the father. The mother being black contributes to that no change in the male-female ratio a little bit more than the father being black. It is known that it's been published and it's been observed for 30 years that the father being black does contribute to a lower number of males. That's been known, but that's not about preterm. That's just about all births. So what does all this mean? Here are the take-home points. So there's no doubt that the male-female ratio is substantially larger than 1 for preterm groups in all race and ethnic groups. There's no doubt about that, except blacks. We see that. And it's fairly constant. This pattern is constant across all these different strata that we've looked at. We're in the process of doing hazard ratios, just adjust for some of these things, but I'm pretty sure nothing's going to sort of tweak these. Usually when you see tweakings in data, it's based on stratified kind of scenarios. These data are built on the base of a very sizable analytic set. So that's what we see, but what does it suggest? So here's where I'm going to get a little bit into the musing. So stay with me for a second, because I have to think about this. It sort of rattles my head every once in a while. So my Bayesian prior would be that what I'm about to say would be very low. That black male fetuses more likely survive and are counted in these earlier gestational ages. So that is, they're not culled before they get to the 20 or 22 weeks. They get over that period of time, and so they count more. So that would make the ratio come down. I doubt it. Black male fetuses destined to be preterm are lost via miscarriage before detection. That fits with the various theories that exist out there. It really does. Oops. So one of the theories that I work with some economists up at Berkeley who have been theorizing for a variety of things for a while. And this is based on a lot of evolutionary biology, that natural selection has conserved our biology to really sort of make us want to have grandchildren, to invest in grandchildren. He only needs 5 minutes. So the intent on natural selection is to really maximize our fitness, and it's to minimize our investment. Males are a big investment. I don't need to tell all the women in the room that males are a big investment. And mostly because we're more apt to have disease. And that's true for all nations, all years, we're more likely to have disease. So we're going to change to Brice. The mechanisms that we're putting forward is that it could be stress. Ambient stress has been identified as changing the male-female ratio. The 9/11 attack in New York changed the male-female ratio in California. That's been observed. The earthquakes in South America were associated with a reduced male-female ratio. The idea is that the frail males are cold. So now the idea is that another idea is that we're observing infection and inflammation, and this is where Brice comes in. So Brice, come on up. But while he's coming up, no, you don't have to clap. Clap afterward. Clap what? But I do want to make sure that people know that David Stevenson, Jonathan Mayo, Mike Eisenberg, Virginia, Brice, Nima, and my colleagues up at Berkeley have been involved in this work. So, out. Younger and better looking colleague, but forget that.
Okay, so I have 42 slides to go, so let's see if I can do that. 7 minutes, perfect. 7 minutes. So the good news is I'm not going to talk about mechanisms because we don't have them yet. But really the goal here is to introduce you to the tools and methods and analytical methods that we're using to get to these mechanisms and understanding sex differences during pregnancy, both from the maternal standpoint and from the fetal standpoint in humans. So our group is primarily interested in understanding the maternal immune response to a healthy pregnancy so that we can then identify deviations from normal that are associated with abnormal pregnancy, preeclampsia, and preterm birth being the pathologies that we're mostly interested in. Why do we focus on inflammation and the immune response? It should be obvious by now. The maintenance of a healthy pregnancy relies on a finely tuned immune balance, okay, from mechanisms of fetal-maternal tolerance that are initiated early at the beginning of pregnancy and maintained throughout pregnancy for exactly 9 months. And then these mechanisms need to be counterbalanced by usually the revving up of the innate immune response so that the host can be protected against invasive pathogens, for example. Now, there's emerging data that there are sex differences in these immune responses, and we've heard from Virginia Nguyen and the prior talks. The consensus, or most of the studies, are suggesting that a male pregnancy is traditionally associated with increased pro-inflammatory responses, but by no means this is set in stone. And there's a nice study from a group in Cincinnati who's showing now that in female pregnancies, if you stimulate the cells right, your peripheral immune cells right, and look at at cytokine responses, you're seeing increasing pro-inflammatory cytokine responses in female pregnancies. So there's many ways that one can measure inflammation and study inflammation, and certainly prior studies have produced tremendous amount of data using transcriptomic approach, proteomic profiles, and this is important. One of the limitations of these studies is that those are studies that are done on using bulk analyses of pooled immune cells, or looking at secretion of proteins in the plasma. And what they're lacking is a single cell resolution. And this is important because your immune system is made of multiple immune cells, and if you start looking with a single cell approach to it, you see that cells are doing different things. And so a pooled analysis may not really reflect what is happening at the level of individual immune cells. So we take a slightly different approach. We're interested in understanding how immune cells themselves, each one of them, is responding to pregnancy what are they doing in terms of phenotypic changes, distribution, and functional changes, and hopefully understand mechanisms that differentiate male versus female pregnancies. And that'll be important when we try to understand mechanisms that maybe predispose male pregnancies to preterm delivery. So one way that we do this, that's our weapon of choice, it's called mass cytometry, and it's a high-dimensional flow cytometry platform. It was developed in Gary's lab, Gary Nolan's lab here at Stanford. And what it does is, if you guys are comfortable with flow cytometry, it's the same principle, except that for every one of every immunological cells present in the blood samples, we can measure as many as 50 parameters per cell. So some of these parameters will allow us to measure surface markers, but we can also measure intracellular parameters. And I want to give you here an example of the type of data set that we generate using this method. And you'll see that we're moving, away from, the immune system being separated between innate immune cells on one hand and adaptive immune cells on the other hand. And we can represent them a little differently. So with 50 parameters, we can first define our immunological landscape here. Those are all the immune cells present in your blood samples, and you can be as fancy as you want. Here, we're just using a traditional gating strategy. So now the orange dots here represents the distribution or the frequency of these immune cell subsets laid out on using this graphical representation. Now, in every one of these immune cell subsets, we can then measure how these cells are functional. And there's many different attributes that we can measure. We can measure signaling responses, cytokine response, RNA transcript. Oh, I'm almost done. And then we can look at them at baseline and also how they respond to their environment, so by stimulating these cells. And the bottom line here is that we can then visualize the immune system as a intercorrelated network of immunological events. Okay, there's a large complexity in this type of data set. It's high dimensional, it's correlated, and it's multi-layered. And so we rely on bioinformaticians. We work very closely with Nima Gaipor's group to then identify predictive features that are associated with an outcome of interest, male-female being one of them. So we've applied these technologies recently to develop a model that shows that by measuring immune cell response in the periphery, we can actually accurately predict the gestational age of pregnancy with p-value of 10 to the -16. That's not bad. And then most recently, we've actually used this system to differentiate normal pregnancies from pathological pregnancy here in preeclampsia and build multivariate models that accurately predict preeclampsia based on a blood test that is performed much earlier than when the symptoms are actually happening. This is work from Shayuan and Sajad in my lab, and in Nima's lab. So the point here is to use these methods to now get to female and male differences. And so I want to show you, this is really, it's premature, no pun intended, premature data, preliminary data from Laura Peterson, who's a neonatal fellow in the lab. And she's actually looking at the other side of the spectrum. She's looking at cord blood. Her major project is to understand age differences in immune development in cord blood using this technology, but we have the data, so we can look at male-female differences. Only 4 female and 6 males, okay? So I'm not going to go into interpretation here. But the bottom line is that she took this systems-wide approach, looking at every one of these immune cell subsets defined in cord blood, and look at male differences. And there's already very interesting differences that are appearing, differences in distribution of plasmacytoid dendritic cells between male and female, decreased Treg frequency in male, and then increased type 1 response to interferon, like a viral type of response, increased inflammatory response in male cord blood. So this is preliminary data, but I think it's going to be very exciting to increase our sample size and start getting at the mechanisms. That's it. 30 seconds late.